What drew me to ERDC was the people here. The people here are so enthusiastic about science and being part of the mission to help the nation.
The really interesting problems that we get to work on and the amazing people that I get to work with.
I like a challenge and I like change. And it was pretty obvious to me upon learning about ERDC that there was a lot of really unique challenges, and it was a dynamic changing environment that had a lot of really meaningful impacts for both our civilian and military sectors, and that really appeals to me.
Was knowing the fact that I get a chance to be a part of a worldwide network of bright researchers and scientists that ultimately get a chance to help improve our environment as well as our technology.
It helped the military, it helped our nation, but it also helped different local individuals in different communities that not necessarily can help themselves. So I'm humbled to say that I'm a small part of this community that's providing this help to our nation.
I think the biggest benefit that working at ERDC has is the ability to choose your own adventure.
This is public service at its best, if you will. You're working at a government lab and serving the country, the people, the armed forces. And I think that, to me, that's a great motivator.
So if somebody were saying, hey, should I come work for ERDC? I would say yes. Yes, you should. Absolutely.
Basic research is critical for the future of the Army. If we solve today's problems, we will fail in tomorrow's battlefield. So we have to be able to forward project possible new futures. Dynamic Mutual Information Networks is a project related to network science. Okay, network communications, networking of our logistics, networks that we understand, data transmission networks is something that will be with us for the foreseeable future. So developing new techniques that allow us to understand, adapt, and disrupt these networks will be critical for the success of future Army operations.
This project delivers fundamental knowledge toward creating a more resilient network through bio-inspired solutions to enable future Army capability beyond current operational standards. Our research explores the role of non-native birds in ecosystem functioning on Oahu, where we find that non-native birds are almost entirely responsible for native plant dispersal on the island. This research was the first of its kind to document how species interact in a novel ecosystem using a network science approach. The developed frameworks have the potential to inform other military relevant issues, such as tactic, techniques, and procedures, TTPs, relevant to insurgency and counterinsurgency operations.
So we can take the math that Dr. Sperry's team has learned by examining where do birds eat in Hawaii, and then where do those plants grow once they've been eaten, and take that and use it as a network that takes information, and then the Army can take that information and transfer it somewhere else, or we can take those networks and disrupt them, or we can establish new supply lines that will allow us to continue to feed and water our troops the same way that we fed and water our bushes. ERDC's basic research is focused on solving the impossible problems of our future battle space by anticipating the Army's challenges to come. There are no new problems, there are just new ways to solve them, and that's what we invest in, is a new future here by rewarding high-risk, high-reward research.
Welcome to ERDC Live. I'm your host, Shelley Tingle with the U.S. Army Engineer Research and Development Center. Today, our special guest is Dr. Jinelle Sperry, a wildlife biologist with ERDC's Construction Engineering Research Laboratory and lead of ERDC's Threatened and Endangered Species Program. The research conducted by Dr. Sperry and her team is at the forefront of supporting the Department of Defense's efforts to better understand where threatened and endangered species are located on DoD lands and how to better protect and preserve these critical ecosystems. This work also ensures our military forces can conduct crucial training operations while drastically reducing the impact to any species in the region. The R&D conducted by Dr. Sperry and her team and the deployment of state-of-the-art technologies has made the process of tracking, quantifying, and protecting these species easier and faster and less expensive. The research was also featured in the recent edition of the Power of R&D newsletter, a quarterly publication that highlights the latest R&D efforts within the U.S. Army Corps of Engineers. Dr. Sperry, welcome, and thank you for joining us today all the way from Washington, D.C.
Yes, thank you so much, Shelley, and thanks for the introduction, and I'm so happy to be here with you today. Well, remotely at least.
Remotely, I love it. And so tell us a little bit about your journey. Like, how did you become interested in being a research wildlife biologist and your journey with ERDC?
Thanks. Oh, gosh, where to start? So I've always been interested in nature and wildlife. I grew up in Montana, a beautiful area with lots of beautiful natural areas. So I did my undergraduate in wildlife biology at University of Montana, and then my master's in wildlife at Humboldt State University in California. And then I came to University of Illinois for my PhD, and that's where I became familiar with ERDC, but also the U.S. Army mission and
conservation of wildlife species on those lands. So my PhD research actually took place on Fort Hood, Texas, studying predator-prey interactions between snakes and 2 endangered bird species. And that work, my PhD was actually funded through ERDC, and so that was how I became familiar with kind of this whole, all of ERDC and the Army. And so then I became a federal employee at CERL at the Construction Engineering Research Laboratory in 2010. And since that time, I've been, kind of the de facto lead of the Threat Endangered Species Research Program here at CERL and ERDC.
That is so fascinating. I always am very interested in people's ERDC journey and how they got here. So I want to remind our audience, I don't want you to forget that you are part of the conversation. And so please ask Dr. Sperry questions right there in the chat box. I will be sure to ask her in just a few moments. So Dr. Sperry, tell us a little bit about what is your Threatened and Endangered Species, or TES program, and why is it important?
Yeah, I get that question a lot when I say that I'm an Army wildlife biologist, and people say, why does the Army even have wildlife biologists? Well, it's largely because the DoD and the Army, as landowners, have threatened endangered species on their lands. So actually, the DoD in general, and then Army specifically, has the highest density of threatened endangered species of any other federal land agency. And that's largely because these installations are often islands of habitat, kind of in a sea of development. And so we have really large numbers of threatened endangered species on these lands. And of course, that can potentially be in conflict with the military mission. So the presence of that species can be costly for the Army, both in terms of managing the species, but probably more importantly in terms of training restrictions. And so if a species is present on an installation, that may result in restrictions such as whole areas of an installation that are off limits to military training, or maybe more challenging where something like a landing strip can be closed off just because of the presence of a species moving through. And so our work at ERDC is we call it science to solution. So we try to develop the science that can then be put on place on these installations to more effectively manage and conserve endangered species, ultimately so that we can improve the military mission and allow flexibility for military training. So Fort Hood is actually a great example of this. So I said that I did my PhD work at Fort Hood. I still am doing research there. I've been doing research there now for over 20 years. And so prior to 2005, about 55,000 acres of Fort Hood were restricted to military training due to the presence of 2 endangered bird species, the black-capped vireo and the golden cheek warbler. And so, but Fort Hood and ERDC teamed to be able to assess how those species are doing on the installation and kind of make the argument to the Fish and Wildlife Service, which is the regulatory agency for the Endangered Species Act, for why the species are doing well there and why we should have training restrictions eased. So in 2010, following these analyses, Fish and Wildlife Service reduced the number of acreage that was restricted down to less than 10,000. And in 2015, actually all of those lands were opened up for training. So no more restrictions on the installation based on the presence of those species. Actually, and in 2018, one of those species was actually delisted from the Endangered Species Act in large part because of the successes at Fort Hood. So this provides this example of this win-win situation where the species are doing well and the installation has more flexibility in military training than it's had in decades. So this is kind of the goal of the Endangered Species Program at ERDC is to allow these species to thrive on the installations while simultaneously fulfilling the Army's mission.
Absolutely. And I like what you said. It is a win-win for our installations, and it's a win-win for endangered species. And so can you tell us about some of your recent research about the role of your invasive species on seed dispersal, specifically the seed dispersal networks? Why is this research important? And can you tell us a little bit about the seed dispersal network?
Oh, I could tell you a lot. So Shelley, you just give me the hook when it's when it's time for me to stop talking. I could talk about this all day. So most of my work is actually focused on community ecology. So instead of studying individual species, I study species interactions and how that influences conservation and management of species. And so a lot of my previous work had been on predator-prey interactions. I said that about Fort Hood. But in the last 10 years or so, my work has focused more on plant-animal interactions. And so a lot of this work takes place in Hawaii. And so Hawaii is kind of an interesting case. There's several installations, military installations out in Hawaii, so it's important for the military mission. But it's also referred to as the invasion and extinction capitals of the world. So there's been a really large number of extinctions of the native wildlife species, but there's concurrently invasion of all of these non-native species. And so this can influence, of course, the species themselves, but it also influences the interactions between species. So so Hawaii is interesting because about 80% of the native plants in Hawaii are adapted for bird dispersal. So the only native mammals on Hawaii were bats. There were no native rodents, and so the primary dispersers of fruits were birds historically. And most of these species actually require bird dispersal. So the seeds actually have to go through the gut for germination to occur. And so the vast majority of these species across the Hawaiian archipelago, these plant species are dependent on bird dispersal. But almost all of the native frugivores, that's animals that eat fruit. So most of the native frugivore birds are extinct. In fact, there's really only one remaining across the Hawaiian archipelago in any decent numbers. And there's none on Oahu. So all native frugivore birds are all extinct on the island. But there's a lot of non-native birds. And so we were exploring whether or not these non-native birds are serving the ecosystem role of dispersing native plants, these endangered native plants that the Army is managing. And so we've been out there for 7 years now. So we set up nets and we catch birds, and they're nice enough to defecate into a paper bag for us so that we can sift through that fecal sample and see if there's seeds in that sample and see what type of seeds they're eating and if those seeds are viable. And the results were really surprising. So we found that 100% of interactions were novel in that we didn't have a single interaction between a native bird and a native plant. We did have some of these non-native birds eating native plants, which we were somewhat excited about, except for about 96% of interactions were with non-native plants. So really, these invasive birds are moving invasive plants. And this is kind of this quintessential, this invasional meltdown, where you have non-native species encouraging non-native species. So you have kind of this runaway train of invasion. But it's important to understand what's going on so that we can effectively manage. So this work seems to indicate that even though the non-native birds are kind of the only hope for these native species, they really predominantly eat non-native plant species. And so now we're working on trying to develop management that can help these native species based on this key information that we needed that we didn't have before this work. And so we're doing a couple of things. One is that we found that that these birds, if they're in an area with the native plants, that they will actually eat them. And so we've been doing playback. So we play calls of these non-native birds, and the non-native birds will come into the area where the Army has been doing restoration on these native plants, and they'll actually eat the plants if you can get them to come in. So we're doing playback to try to get these birds to come into the sites. We're also working at removing some of these invasive species to try to get the native species, the non-native birds, to eat more of these native plants. And so that type of work, I think, is what's needed kind of in a variety of systems, is understanding not only the individual species themselves, but these really critical interactions that really make the difference of whether or not we can successfully conserve and manage endangered species.
That is just incredible. Learning how sometimes doing research, you find things out that you didn't expect. I mean, that's the whole point of research is acquiring new knowledge. And so that is absolutely fascinating. So how are you leveraging new technologies such as eDNA. And for those that don't know about eDNA, do you want to explain a little bit about eDNA?
I love to talk about eDNA. So eDNA is, anyone can attest who's ever talked to me. So eDNA is environmental DNA. So instead of sampling for animals or plants directly, such as trapping them or doing visual counts, instead we can just sample for their DNA. So plants, animals in the environment, actually you and I sitting here right now are sloughing DNA into our environment. And so we can collect that environmental sample. So for a terrestrial animal, that can be soil. For an aquatic species, that can be water. You can even collect DNA from the air and know what species are present in that system. And so this is a technology that's really emerging. And I think it's going to be a game changer for the way that we survey and manage wildlife species. And so, gosh, probably 5 years ago, my work was probably 80% using traditional surveys and probably 20% using molecular. And then the last couple of years, that's flipped. The majority of my work now uses molecular tools just because we found that it can be so much more cost effective for surveying for these species compared to traditional survey techniques. So a couple of examples of this are, we just wrapped up a project with alligator snapping turtles. So alligator snapping turtles, if people are familiar, are these very large aquatic species that are found in the southeast. Actually, those from Vicksburg are probably very familiar with an alligator snapping turtle. And they were recently proposed for federal listing under the Endangered Species Act. But they're really difficult to sample for. So currently the way that we sample for them is we put out these big hoop traps in the water. We bait them with sardines or cat food. And then we go back every day and check the traps. And if there's a turtle, then we take the turtle out and we mark them and let them go. But it's dangerous for the turtle. It's dangerous for the technician. These can be pretty aggressive species. And it's incredibly time intensive and expensive. Instead, we can just go out and collect some water and know whether or not the DNA is present in the system. So we've been doing this work at Fort Polk, Louisiana, where we'd been doing traditional trapping, and we had, over 6 years, I think, we had trapped 3 streams. It's just too difficult to be able to trap at a large area. We went out and collected DNA. We collected at 30 streams in a weekend across a couple of days. And we could sample across the entire installation. So we were able to really increase information about where that species occurs on the installation, and maybe even more importantly in terms of Army mission, where the species doesn't occur on the installation, so that we can start to get an idea of their distribution. And we found that the costs of eDNA compared to traditional sampling can be 2 to up to 70 times less expensive using eDNA compared to traditional, as you can imagine. But we could also use eDNA to assess communities. So for single species, I'm going to get nerdy on you for a minute. For single species, we're looking for a DNA sequence that's unique to one particular species. That sequence that's unique to alligator snapping turtles. And what's funny is, so we use PCR, and so before COVID, that was not a familiar term. I think now it's just kind of in our common vernacular about what PCR is. So instead of doing a nose swab and looking for a sequence that's unique to COVID, instead we collect a water sample and look for a sequence that's unique to alligator snapping turtles. We use the exact same technology as PCR is what they use for COVID testing, which I will tell you made it very difficult to acquire supplies the last couple of years. They're in pretty high demand right now. But you can also, instead of looking for one particular sequence, you can actually sequence all of the DNA that's present in a sample, compare that to all known DNA sequences, and get at the entire community that's present. So we're doing this at a variety of installations around the U.S., Fort Hood, Fort Polk, Fort McCoy, Fort Belvoir, kind of all over the U.S., where we're looking at whole communities, for example, fish communities, where we can go in and collect water. At some of these sites, we find that in a single sampling effort, we were detecting 70 to 80% of all of the fish that had ever been detected on the installation via, fishing and electrofishing and all of that. we can do it in just a few days of sampling. So the potential is incredible for both identifying single species and kind of getting distributions of those single species, but also looking at my interests, looking at community ecology, looking at ecological communities, what species are present in a system, and understanding potentially how those species interact.
So you can look at things, because of all this data, you can look at things at a macro level and a micro level of understanding your whole ecology system.
Exactly, and with the same sample. So at Fort McCoy, we collected one series of water samples, and we've looked at the fish community, we've looked at the plant community, we've looked at the invertebrate community. So you can look at, instead of, and if you can imagine traditional sampling, for example, invertebrates, so like insects, requires a lot of taxonomic expertise. The difference between these species often comes down to these really minute morphological characteristics. But for DNA, it's just the DNA. And so I can identify down to the species level just based on that DNA sequence, not based on morphology. And so you don't need all of that taxonomic expertise for identification. So instead, you can spend your time and effort looking at, entire communities just based on a single water sample. So again, kind of the possibilities are endless when you start to think about the kind of quality and quantity of data that you can capture in relatively low effort.
And how data collection has changed just in your career.
Yes, I was actually, that you're getting older when you sit there and reminisce about the good old days, but I was just actually meeting with someone that I was a field technician with, 20-some years ago, and we were talking about, we didn't have GPS units
That's right.
when we first started this work, and now you can sequence, entire communities that are present in a sample. Yeah, the technology is really changed the way that we conduct research for wildlife.
That's just absolutely incredible. And so thinking about EDNA and how you have completely changed the way you collect information, can this be applied to things like, can it be used for the mitigation effects of changing or a warming climate? Can it be used for those type of problems?
Yeah, that's a great question. Yeah, I think that eDNA has the potential to address a lot of these really sticky, difficult challenges that we face. In many cases, for example, for climate change or a lot of these challenges, it's difficult to be able to address these because we don't have just that basic underlying information on some of these species. And so, and that's often because it's really difficult to acquire the type of data, even like baseline data on species distribution, much less understanding physiological tolerances and all of those things that go into predicting and mitigating impacts of climate change. And part of the reason that we don't have all of the data on every species is because there's limited resources for conservation and for management. And so I think it ends up being a bit of a triage. And so we spend a lot of our resources towards these critically endangered species. And then there's just not enough resources to be able to get even baseline information for a lot of other species. And so eDNA allows us to be able to capture data for a large number of species for a relatively low cost. And to be able to start to get to those questions. If we understand distribution of species and where species occur, then we can start to understand physiological tolerances and understand how some of these abiotic factors and how climate may influence these species. Again, something that's of great interest to me is species interactions. So understanding which species are present in a community can help us to understand how those species may interact in the face of a changing climate. So for example, a lot of our work in Hawaii has been based on networks and network ecology. So understanding how these species interact in these networks, particularly when we're talking about interactions that are critical for these species. So if one of their partners are not present, then you can start to see that whole system collapsing. And so eDNA, I think kind of the next generation of eDNA, is being able to understand, these communities, understanding networks and how these species are connected kind of across space and time, so that we can start to be able to predict how these changing environments would influence not only individual species, but also how these species interact in kind of these networks of individuals for conserving, again, not just that critically endangered species, but conserving and managing, entire ecological communities.
And so I want to remind our viewers that we are taking questions from you. And if you just joined us, I am interviewing Dr. Jinelle Sperry, a wildlife biologist with ERDC, and we are discussing the Department of Defense's efforts to better understand where threatened and endangered species are located on DoD facilities. And so right now I'm going to go to our first audience question, and it is, what are the future uses of eDNA for USACE or other agencies?
Yeah, great question. So I think I touched on this a little bit, but I think at this point it feels like the sky's the limit for eDNA in terms of future use both within the USACE and then outside. This has just been an exploding technology, because I think as people kind of understand potential uses for it, then they're using it, and then we also find new uses. So some examples are the use of eDNA for detecting invasive species. So this has been an area that eDNA was originally used pretty heavily for. This was aquatic invasive species. eDNA was used to detect aquatic invasive species. And so I think there's still potential there in understanding kind of invasion and monitoring areas for the invasion of these non-native invasive species using eDNA, again, as kind of a cost-effective way to survey large areas. But then we can also use, as I discussed, eDNA for understanding distributions in communities that have at-risk or endangered species. But then I think the next step is kind of going beyond the just presence absence, just understanding whether or not species are present in a system, and instead using eDNA to look at the ecology of a system, and understanding, as the question earlier about how things like climate change can influence these communities. So for example, we're just working on a project right now where we're looking at not only the presence of trout, but we're looking at the presence of trout and one of their main parasites, a gill lice. And so we can understand where the fish species and where their parasites occur, and we can overlay that with other information such as other invertebrates or abiotic factors or habitats to understand kind of parasite interactions with these species to try to understand more about the ecology. So I think that's kind of the next step of eDNA. I also think that there's a big push to not only understanding presence absence, but also trying to use eDNA to look at abundances of individuals that are present in a system. Right now, I feel like this is a bit of the holy grail of eDNA, about trying to understand not if something's present, but how many are there. Because I think that has been a bit of an issue with eDNA to date, is we can say that there's potentially a species there, but we don't know how many there are. We don't know if they have any diseases. We don't know if body condition, etc. So as we start to improve these eDNA, we can start to look at, what species are there, potential relative abundances, and then look at their potential parasites and or pathogens to try to understand more the ecology of these species as opposed to just presence absence.
So there's a lot more research that needs to be done.
I think we're just scraping the surface.
So it'll be interesting when we have this conversation like 5 or 10 years from now of how it's evolved. So another question we have is from Brent, and he wants to know, he said, Dr. Sperry's work, has any of your work been related to the Army Capable Use Buffer Program. So has any of your work been related to the Army Compatible Use Buffer Program?
Yeah, so ACUB, Army Compatible Use Buffer, that's a great question. ACUB is a program in terms of endangered species. This is often where an installation will be able to manage species outside of the Army lands themselves. So that instead of managing species kind of in perpetual conflict with Army training, instead we're managing them outside of the installation boundaries on partner lands, for example, such as nature conservancy or such, where we can manage those species away from Army conflict. So actually all of my work in Hawaii takes place actually off of the installations. It's kind of a combination of ACUB and then some other programs that allow management to occur off the installation. In Hawaii, gosh, I'm actually writing a lot of the ESA consultations, so I should know this off the top of my head. I think Oahu has 72 endangered species that the Army is managing, which is, Fort Hood, a lot of these other species maybe have 1 or 2 endangered species that they're managing. On Hawaii, you're talking dozens of endangered species that Army is managing. And so many of those we're actually managing off of the installation through things like ACUB. So not necessarily, I don't think I've actually done any EDNA work on any ACUB lands, but a lot of our endangered species work is associated with these ACUB properties, which I think is a really effective and smart way for the Army to manage these species, in perpetuity with these partners without having to manage, right on the Army installation lands themselves.
Very
good question.
So our next question is from Dr. Amar. He says, can you still tell the past presence of a species if it has already migrated to another area, even if years earlier? So let me repeat
I love this question.
I just want to make sure you got it. Can you still tell the past presence of a species even if it's already migrated?
I love this question, and I have spent so much of my life the last couple of years trying to address it. The short answer is that DNA degrades, and so depending on kind of the system, the DNA that you're collecting could have been from right then, or it could have been collected from it could have been deposited sometime in the past. And so that's a great question, because we need to understand that if we're coming to example of Fish and Wildlife Service and we say that a species is or is not there, we want to know if it was there yesterday, if it was there 2 years ago. And so there's been a lot of work in this area trying to address this question. Part of the problem is that it's contextual, in that UV, solar radiation, degrades DNA very rapidly. So for example, if it's a system that if you're in a pond with full sun and you're right on the surface of the water, that DNA probably only lasts an hour or 2. If you're talking about underneath the soil, the DNA can last a lot longer. I mean, if you're talking, we also know that there's historic DNA, very old DNA, and things like caves, we can find really old DNA. So depending on kind of the system, depends on how long that DNA is present. We're actually doing a project that I'm really excited about right now, where we have I've done work with Louisiana pine snakes. So this is do I have time to talk about this quickly,
yes,
Shelley?
Absolutely. Talk about pine snakes.
Okay, it's one of my favorite topics, but so Louisiana pine snakes, this is an endangered species. It was listed in 2018. It's Fish and Wildlife Service considers it one of the most rare vertebrates in North America. It's currently really only in 4 small populations across the southeastern U.S., and one of those populations is Fort Polk, Louisiana. So there's real potential for it to influence training, because this is a terrestrial species that often is in areas where training occurs, and so it has the potential to lead to training restrictions. And so there was a lot of interest in the species. How we typically collect or get data on this species is trapping, and so they have a really extensive trapping program on the installation and outside, where they set up dozens of traps, and again, check them every day to see if we catch snakes. This represents, every year, it represents thousands of trap nights. So nights per each trap being out. So every year that represents thousands of trap nights that they put effort towards trying to catch these snakes. And on average, they catch 2 snakes a year on Fort Polk. So hundreds and hundreds and hundreds of man hours for 2 snakes on average a year. Many years they don't catch a single snake. And so you can imagine that this is not a cost-effective way to survey for this species. The species spends about 80% of its time underground in gopher burrows. And so we thought, well, what if we could just collect the soil of the gopher burrow? Because we know that it's underground, and so that DNA likely persists for a while, and that the snakes are kind of congregated in these burrows. So we'd be more likely to actually be able to get DNA. And so we started that project. We developed an assay, that single sequence that's unique to Louisiana pine snakes. And we started collecting soil, and we found that if we collected right where we knew a snake was, we could get DNA, we could detect DNA 100% of the time. If we just went to a burrow where we know in general snakes were, we could detect DNA 50% of the time, which is a huge improvement over trapping. But then the next question that we get often is, well, what does that mean? How long ago was the snake in that burrow? And so we started an experiment in our thermal chambers where we collected a bunch of soil, we put it in tubes, and then we half the tubes we exposed to high UV, and then the other half we exposed to low UV, and then half to high temperature, half to low temperature, and then half to high water input, thinking that the water would just kind of soak the DNA through, and then half to low input. And so this is hot off the presses, we just got the results back. And the DNA, after about 2 months, the DNA degrades by about 80%. So I think we can, in a terrestrial system, we can say that DNA lasts, you can get small fragments after that, but about 2 months in that system. But again, so context dependent. But again, as we start to, this is the critical research that we need to be able to really use this type of data for compliance, is to know what does it mean when we get a hit. So in this case, for Louisiana pine snakes, we can understand if we detect Louisiana pine snakes, chances are they were there in the last 2 months.
And that's information you didn't even have until recently. And so there again goes into the importance of basic research and the quest for knowledge and understanding the whole problem. And so our next question from our audience member is, are installations, including EDNA research for species in their INRMPs, are they able to secure funding at the installation level through the Sykes Act?
So that's the Installation Natural Resources Management Plan, is the INRAMP. And so this is, just for those who aren't familiar.
Yes, absolutely. Spell out those acronyms for us.
I'm a government employee. I know all the acronyms. So the INRAMP this is the management plan that the installation makes for all of their natural resources and endangered species on their installation. There's also an installation management, endangered species management plan. But these go hand in hand, so that the installations are really thinking about their management and how that's going to influence natural resources and endangered species on their installation. And a benefit of the INRAMP is if these installations have an INRAMP that's approved both by the installation and by Fish and Wildlife Service, then they will not have Endangered Species Act critical habitat assigned on their installation for these endangered species. So this is a real key aspect of consultation for the Army. And so these INRAMPs are really critical, both in terms of planning for the installation, but also in terms of Endangered Species Act compliance. And so, yes, the short answer to that question is that many of the installations that we work with have been funded funded through the Army to use EDNA for surveying for endangered species. So this is true on, I think we're on 20 installations. My team is working on around 20 installations right now. Many of those are using EDNA. And so I think at this point, a lot of these are somewhat pilot studies to try to understand the use of eDNA and if that is a viable tool for them. As we do more of this research, understanding what it means to get a hit, all these things that we've been talking about this whole conversation, I think then we can start actually using that eDNA in ESA consultation process between installations and Fish and Wildlife Service. But at this point, yes, the data that we've been collecting using eDNA has been included in several installations in ramps that are ongoing. So the answer is yes, and I think as this technology matures, I think we're going to see it more and more in use on these installations and with consultation.
Very good. So our next question is about the abundance of all this data that you're bringing in that you've never had before. And so what is needed to make eDNA research more successful, like the use of data analytics?
Yeah. It's difficult because eDNA right now requires a lot of very expensive laboratory equipment and tools, and then once we actually get, like, for example, sequence data, we end up with billions of reads of sequence data. So we often need very complex bioinformatics to be able to get that data into something useful. And then oftentimes we require supercomputers to be able to actually analyze that data. And so at this point right now, this requires an installation, for example, or a district, to team with others. So to team with laboratories, ERDC, etc., that have these equipment and bioinformatic tools and supercomputers to be able to analyze all of this data. But again, the technology is moving very quickly. There's even new backpack eDNA contraptions where you can go out into the field and collect DNA in your backpack and start to get some information on species occurrence. So I think as technology advances, as we just talked about GPS units, as technology advances, I think we're going to be at the point where we can get eDNA into the hands of the actual practitioners, into the hands of the biologists who are on the ground doing the work, where you don't need all of that background equipment and computing power. But at this point, we do. So at this point, it requires, again, often collaborations across numerous partners to be able to pull all these pieces together.
And so our next question from an audience member is, could eDNA be used to focus on an individual organism, such as marking an individual organism without having to capture the species at all?
So, I'm sorry, can you repeat that question?
Yes.
Make sure.
Could eDNA be focused for an individual organism, such as marking an individual without having to capture the species at all?
Yeah, so we wouldn't be able to necessarily, because we're just collecting their DNA, and then that DNA is no longer, attached or part of that animal. We can't mark an individual, but I think maybe what the question is can we actually get at individuals? Yes. And so when we're using eDNA, we're looking at just a really small fragment of DNA, 150 base pairs, really small little fragments of DNA for a particular species. And we often look at particular genes, and those genes are often genes that are associated with, for example, metabolism, that are highly divergent among species, so that sequence ends up being different on that species compared to another species, but not necessarily individual. We can, we know this from CSI, right? That we can collect DNA and get information on individuals, but these are much larger fragments of DNA to be able to actually get at individuals. And so that is often, usually the DNA that we collect in the environment is really kind of chopped up, and so we don't have these really long fragments. And so from something like tissue, we could get at individual. From DNA, EDNA, we're often looking at kind of smaller fragments. But part of it comes down to cost too. Right now, it's really expensive to be able to sequence really, really large fragments. Over time, that I think is going to get cheaper, where we can, again, start to use eDNA to look at individuals, look at population genetics. We're very close. Some would argue maybe that we're actually already there. I think we're very close, but right now it's not quite of that cost benefit that we see with eDNA when we're looking for these little tiny fragments that are unique just to particular species.
So our next question is an article in a recent USACE R&D newsletter discusses how your research is applied to USACE challenges with invasive carp. Can you tell us more about this research?
Yeah, so this isn't my research, but there's been really amazing research done across the ERDC looking at the use of eDNA for Asian carp. And a lot of this work was done by the environmental lab down in Vicksburg. But so Asian carp, or invasive carp, I'm sure most people are familiar with this story, are invasive species that's been slowly moving up our waterways. There's real concern if the carp get into the Great Lakes. And so the use of eDNA for invasive carp is really kind of one of our really early examples of how eDNA can be used really effectively. And so there are these really large monitoring efforts going, across many states and many waterways, using eDNA to more cost effectively look across really large areas for potential invasion of an invasive species in this case. And so I think eDNA, that's been one of the early adopters. USACE was one of the early adopters of this technology for invasive carp. And again, now it's being used in a variety of contexts. But I think that use, eDNA to detect invasive species, is a really good example of how eDNA can be used to more cost-effectively manage natural systems. So, for example, at Fort McCoy, Wisconsin, they've asked us to start a long-term monitoring program where we go to these stream and lake sites on Fort McCoy every year and do eDNA monitoring so that over time we can start to see potential invasions. So whatever your invasive species of choice, let's say invasive carp, we don't have it, oh, we got it, then you can run in and do early removal or early management, so that we can start to manage these species before they become a problem. Again, there's limited resources and limited dollars, and so it's hard to, using traditional tools, to monitor for such a large number of invasive species across a really large area. But using eDNA allows us to monitor across across the U.S. using these tools. And there's started to be, including work done by ERDC biologists, starting to make these large databases of eDNA data publicly available so that people can kind of start to sift through all of this data to document these types of things, document invasions, document presence of endangered species, etc. So I think that Asian carp example, invasive carp example, is a good one of understanding this early detection using the presence of DNA.
And so another question we have from an audience member is, what other questions does eDNA answer?
Yeah, gosh, we've been talking about this, so eDNA, I think, can be used in a variety of ways. So I think eDNA can be used for understanding distribution, presence, absence of individual species. So where an individual alligator snapping turtle may be, an individual Louisiana pine snake, where that may occur. We're also doing work in the Northeast for freshwater turtles. So there's a large number of freshwater turtles that are proposed for listing, spotted turtle, wood turtle, Blanding's turtles. So we can use eDNA to look for presence, absence, distribution of individual species. Then we can use eDNA to evaluate species interaction. So the example that I gave of the gill lice and the trout, we're also looking at freshwater mussels. Freshwater mussels are a species that's of real conservation concern. It's an at-risk group, and they're difficult to survey for. So I've spent many hours of my life surveying for mussels traditionally, and so how you survey for mussels without eDNA is we did a lot of work in Louisiana. You get down on your hands and knees in the water and you dig through the sand until you feel something hard. You hope that it's a mussel and not a piece of glass or other things that we detect. And so you could pull it up and then try to identify it, and they're very difficult to identify based on morphology. Instead, we can collect DNA and know where they occur, but we can also understand where these freshwater mussels occur, but also where their host fish, they're obligate for these host fish. And so we can understand species interactions based on eDNA. And then again, we can look at whole communities and how communities may differ over space and time, and how they may be interacting. For example, for the Army, where you may see communities in areas where there are more extensive training compared to areas where they're not, to understanding, so that we can start to understand potential impacts of military training. I will say I am often questioned about how military training may influence these endangered species, and we find through eDNA and traditional sampling that in many cases these large swaths of habitat that are provided by these installations create such a net positive that the training on kind of a relatively small area often is the negative impacts are negligible. But again, using eDNA to actually document that can make kind of a boon for the Army of being able to actually document and understand military impacts on these endangered species.
Which is absolutely important. And so how has the partnership and support of CERDP helped advance your research and use of eDNA? And what other partnerships have helped advance this research?
Well, that's a good timing for that question, because I am actually, the reason I am in this hotel room is because I am at the annual CERDP conference in Washington, D.C. right now. CERDP has really been on the forefront of eDNA research for the U.S. They've funded a lot of the work that has helped us to understand and adopt this technology. They've funded a lot of work to help us understand how long DNA persists in a system, understand kind of fate and transport of that DNA. They've funded some of the work that's contributed to these large nationwide databases. So CERDP has really been influential in the use of eDNA. I actually, I'm here in part because I have an ongoing ESTCP project right now, which is, if people don't know, is the sister program to ESTCP, to CERDP, where I'm looking at the use of eDNA for documenting pollinators, and I can't believe that I've forgotten to mention this until right now. Because it's a project that's near and dear to my heart right now. So for eDNA, instead of just collecting the water or the soil or the air, we can instead collect actually living samples. So for example, pollinators, I'm sure people have heard about this because it's all over the national news that pollinators are in decline globally. Right. And so this has implications for food security and conservation, but also has implications for endangered species on military lands. There's a lot of endangered pollinators on military lands, but they're really difficult to sample for, and they require a lot of taxonomic expertise. And so instead of sampling for the pollinators directly, which is often lethal, they'll use pan traps to collect those pollinators. Instead, we can actually just collect the flower and look at all of the DNA that's present on the flower and know what species have visited. So we know not only which pollinators are in the area, but we can also know which pollinators are visiting that specific plant, so we can look at networks. And so we did this work initially in Wisconsin at Fort McCoy with insect pollinators and plants, and we found that eDNA is extremely effective for being able to document pollinators. And now ESTCP is funding work where we're looking at bat pollinators on agave in the desert Southwest at Fort Huachuca. And then bird pollinators on endangered plants in Hawaii. So we're going to go and swab plants and know what pollinators are visiting them. And so this is, again, kind of on this kind of forefront of using eDNA, not only looking at individual species, but looking at species interactions. And so this is work, yeah, the ESTCP is funding now, again, kind of all the way from the very basic understanding how DNA works to all the way up to now using eDNA in these really novel and interesting, to address kind of novel and interesting questions. So yeah, so SURIDUP and ESTCP has been really influential in the use of eDNA by DoD, actually, and then adopted by many other biologists.
Very good. And so we have another question, and this is from an ERDC researcher. Would ERDC benefit from developing an in-house database for all DNA markers collected across the labs for more complex analysis to address climate change on a large scale?
Actually, that's a great thought. So there are nationwide databases for sequence data. For example, GenBank, where anybody who sequences an organism, they'll often put this on GenBank so that it's publicly available, so that all of us can then use that sequence information to know what species that sequence is assigned to. But an individual, like a database within ERDC, to be able to look at more detailed information about sequences and distribution data and eDNA, I think that's a great idea. And there are a lot of people across ERDC who are using this technology. So great thought. I will follow up on that, because I do think that is this kind of public access or databases, instead of all of us kind of toiling in obscurity in our own labs, to be able to address bigger questions using bigger data sets, I think is a great idea. Yeah, great question. I love it.
And so this is from Amanda, one of our audience members. If someone wanted to help contribute or even join your research effort, who should they get in touch with?
Call me anytime. Yeah, we're always happy to have help. Yeah, we have. So I didn't mention this, but I'm also faculty at the university. So I advise graduate students through the University of Illinois, and undergraduates. So I have a large number of federal teammates that assist me with my work. Everything from kind of DB4 type researchers, but we also have a lot of technicians. We have technicians embedded at our various installations, kind of seasonal technicians that go to our installations and assist with our work there. We also have a large number of graduate students who work with us, and this is their dissertation work. And then we have between 7 and 8 undergraduates every semester. So yeah, we have a large group of people, and I'm always happy to talk with people who are interested in this type of work. We're spread out around the country doing work on numerous installations, so we're always happy to have help. So yes, if people are interested just in general of either working with our team or learning more about this, this is probably obvious. I'm very happy to talk about this.
Absolutely. And let me throw out a great email for people to use, and it's ERDCinfo, ERDCinfo @usace.army.mil. And so if you want to get in contact with any of our researchers or have even more questions about this topic for Dr. Sperry, we will receive those and will get that information to you after the show. And so we are going to wrap this up, and I want to thank all of our viewers for participating, and obviously thank you to Dr. Sperry for joining us on this great topic. If you want to learn more about ERDC technologies, we also have a fabulous podcast, poweroferdicpodcast.org, that you can listen to about some of our technologies. And so I'll see you next month, and until then, find your joy.